Good morning, everyone. I am Aina P. Roig, Technical Support Supervisor for Clinical Services at Novogene, and I am very pleased to be hosting this Mastermind session as I believe multiomics, having a more holistic approach into the study of complex biological processes, is imperative both in research and clinical studies, and that this concept of multiomics data is revolutionizing the field of medicine and biology. So for this, I'm here with an exceptional lineup with Professor Roig Ewing, Associate Professor in Proteomics and Systems Biology at the University of Southampton, Dr. Ricard Argelaguet, postdoctoral researcher at the EBI, and Yuanyuan Chen, Sales Director from Novogene. So first of all, I would like to invite you all to give some more details on your background. So if you want to begin, Rob, for example.
Sure, yeah. Good morning, everyone. It's a pleasure to be here. So my name is Roig Ewing. I'm at the University of Southampton in the Division of Computational and Systems Biology in the School of Biological Sciences. And, one of the things about multiomics, I think, and the nice thing about this session is that I think it's really timely. I think, we're really looking now at multiomics studies becoming the kind of norm rather than the exception. And I think that's really exciting. We use multiomics, I guess, really to analyze networks in cancer and to understand those networks. And for us, using multiomic techniques, in particular proteomics and RNA-seq together, actually allows us more coverage of those networks and more insight into the mechanisms. So I think, as was just said, when we're thinking about multiomics, we need to appreciate that multiomics allows us to analyze whole systems, and whole systems is really where it's at if we want to understand biology.
Sure. What about you, Ricard?
I've done my PhD at the European Bioinformatics Institute in Cambridge in the UK. And I'm currently working as a postdoctoral scientist at the Babraham Institute also in the UK. So for the last 5 or 6 years, I've been mostly working on the computational aspect of multiomics, because as Rob was saying, like multiomics is now becoming cheaper and cheaper and is becoming a reality for most studies. Now, one of the challenges that is raised from multiomics is how do you actually make sense of these data? Because it gets very, very complex. And then, there has to be underlying statistical methods developed to understand these data. More specifically, I'm more focused on the technological aspect and the methodological aspect of single cell data. So the idea is, we try to get multiomics from one single cell. So is it possible to get chromatin accessibility, DNA methylation, RNA expression information from one individual cell? So this has been mostly the work in my PhD, and I'm happy to take any questions afterwards, and I'm looking forward to this discussion.
Sure.
Okay, my name is Yuanyuan Chen. I am the sales director at Novogene. Novogene is a global NGS service provider. So we provide a platform for the researchers to easily accessible, reliable, and fast, and provide the highest quality. So we have been providing, for example, transcriptomics, metagenomics, and whole genome, whole exome at all different omics level of data sets. So today I can bring from the customer point of view, what are the most frequently asked questions when somebody are in need of multiomics methods.
Okay, thank you everyone for your introduction. So now I would like to throw the first question. So you all mentioned how of paramount importance has multiomics, and clearly this topic is present in different sessions in this Festival of Genomics. So because it generates a lot of interest, but could you give more insight into why multiomics? What kind of questions multiomics can answer while individual omics maybe cannot? So maybe Ricard, you can go on that again.
Yes, I can start. One of the aspects is that any phenotypic change we observe that has a clinical implications, it usually is because it has like some fundamental changes in the molecular profiles of those patients. Now, we typically don't know what are these molecular changes that is happening, and that's why, the more you profile, the more likely you are to spot what is the specific change. And usually in clinical practice, you mostly rely on on single markers to tell if someone has a disease or if someone doesn't have a disease, right? You measure a specific gene, the mutation of a specific marker, and there are many cases where this is just not enough because biology is very complex. And so I'll just give you an example of a case that we were working with a data set of chronic lymphocytic leukemia patients, and there is one mutation which is typically used, to distinguish bad prognosis from good prognosis cases. And for 80% of the cases, fine, you can rely on that mutation, but we found out is that for that specific mutation, even if you have it, there are compensatory mechanisms that allow your phenotype doesn't really express the bad prognosis like challenges. That's why getting from a holistic perspective and pulling information and learning from different molecular layers allows you to get a molecular profile for your patients that is closer to the actual clinical output that you expect.
Mm-hmm. What about you, Rob, in your experience?
Well, I think I can just pick up on something that Ricard mentioned that came to mind there, and that is, absolutely agree with that. If you think about, for example, somatic mutations in tumors, then of course, we're getting to the point where we can profile those tumors more and more easily. But understanding actually what the resulting phenotype is and the actual kind of molecular perturbation that mutation might lead to is more challenging. So, there's been work in the proteomics field to, for example, try and pick up on mutant proteins, right? So if you've got the somatic mutations, you're able to profile those in a tumor. Can you also actually pick up on the mutant protein in your sample? And that actually allows you to distinguish. I think it gives you a lot more information. It gives you a functional readout. And it really, I think, will allow you to distinguish those cases where there is a difference between, for example, patients with a similar mutation profile. So yeah, I think there's going to be a lot to be gained by this kind of multi-layered processing. We've got to remember that biological systems don't respect our sort of rather artificial kind of omics technologies. I mean, most mechanisms are multi-layered, and if we're picking up on one technique to address a problem, we are almost certainly missing a good chunk of actually what's happening in the cell or tissue.
Yeah, definitely. Yeah, from your answers, I mean, we can think really that multiomics is opening a way on how we understand the whole process, right? From genotype to phenotype, which is not that straight line. So it would be providing a bigger picture and more integrative perspective into our biological issues, problems, or things that we want to address. But to put all this data from different omics together, it just needs a very huge effort into carefully considering how we collect, process, and interpret data. So which are the biggest challenges to consider for this data integration and interpretation to have this more comprehensive picture of the underlying biological processes you were mentioning?
Yeah, so maybe I can start here. I think, getting the samples and profiling them is hard, but doing the analysis is even harder than that. And it has been a massive change in biology where most of the time was spent before on doing the experiments. Now, with multiomics, it tends to be the other way around. Making sense of your data tends to be the hardest part. That's probably one of the reasons why it's still hard to use this in clinical practice, like multiomics data, because it's hard to integrate, it's hard to interpret. But methods are starting to come in. And one of the challenges, as I was saying, is the lack of hypothesis sometimes. A patient has a phenotype, but which molecular layer is the driver of that phenotype? Is it changes in the epigenome? Is it changes in the transcriptome? Is it changes in all of them at the same time? So this is the sort of question that like multiomics allows you to answer. Now, one thing I'm actually missing, and I think it's going to be super useful in the future of the multiomics field, is the use of references. Now, because typically when we do an experiment, multiomic experiment, we just work with that experiment on its own, and let's say it's 25 samples. There is a limited amount of information that you can gain from a few amount of samples when you are profiling thousands or millions of features, right? So ideally, what you want is to have some sort of reference maps for each disease that you have, so sort of a multiomics reference map for cancer, for autoimmune disease, for any type of disease. I'm coming from the single cell field, and this is now one of the biggest projects going on in the present, which is called the Human Cell Atlas. So here the idea is to construct a single cell reference map for each organ of your body, so that every new experiment that you profile, the analysis becomes much easier by trying to compare that to the reference. And there has been some work on this on the multiomics field, such as the TCGA, like the Tumor Cancer Genome Atlas, I believe it's called. But much more needs to be developed if we want to take this further into like personalized medicine and clinical practice.
Yeah, definitely feels like more effort into unified repositories for multiomics data need to be done instead of having fragmented everywhere and to connect everything.
Absolutely.
What about you, Rob? So how much we can play with the data we have? How challenging is to integrate all this data, and maybe even how much machine learning on data interpretation can contribute to our progress in these multiomics?
Yeah, so I think a couple of points come to mind. My group is really coming from a proteomics perspective. So we've been interested in interaction proteomics for some time, and we typically use interaction proteomics or quantitative proteomics to solve our issues. But more and more, we want to incorporate other forms of omic type experiments in those workflows. And one of the things we've dealt with for a long time in the proteomics field is really low coverage. So, I think in contrast to many of the other omics techniques, proteomics is perhaps more challenging in a way. So in proteomics, we often have a lot of missing data. We might not pick up on proteins that we should be picking up. We might not have enough coverage of those proteins in terms of their peptides, so we might not be confident about their identification. So, we've had to deal with this issue for a long time, and I think that's an ongoing issue really in the field. How do you actually create samples that can be fully sort of as fully as possible profiled? How do you get accuracy? How much noise is in the data? What happens when you combined multiple data sets together? You might have, is that noise sort of adding from one analysis to another? I think those are questions that need to be addressed. When you bring these data together, are we compounding that noise, or are we actually getting closer to biological reality?
Yeah, that's definitely
just to follow
complicated.
up, sorry, on what Rob said, and what Aina said as well, actually, definitely like machine learning and like data science will be essential to make sense of multiomics data, because it's no longer the case that you can just look at gene by gene. You cannot just look at the expression of one gene, because you have like 100,000 different features that you profile from each patient. So in that case, it becomes essential to pull information across all of these features in a smart manner. And this is done with machine learning, data science, statistics, call it the way you want. But these algorithms, they need data, right? And that's why we essentially need to create all these reference maps. We need to standardize all these different pipelines, all these different repositories to allow us to train these models.
Definitely, No, go Rob, it's fine. I was just only going to say that it's important to have complete data sets and having an extra care, on data quality control, and having the same standards and statistical analysis for all the different data sets. Having said that, I let you go, Rob. Sorry for that.
I suppose I was just going to pose a question actually to Ricard about, if you're going to use sort of machine learning approaches, then, most machine learning approaches need some kind of validated training data. So, what would we think of in terms of the ideal training data kind of in complex biological samples? Is that really are we going to be able to use those for training, or should we be thinking perhaps about synthetic samples?
I mean, one thing one point to think about is that your model is as good as your training data is. So it's definitely a very important question. Like there is no magic out of the box models that will solve this problem. Now, having said that, it's true that there is a lot of technical noise. There's a lot of batch effects. There's a lot of challenges when you work with this data. But again, the more samples you end up profiling, all these different noise effects, they tend to balance out. And using the right algorithms allow you to extract signal that is going to be useful to either predict your specific clinical phenotype or any output that you care about. But the essential turning point for multiomics is getting these large scale data sets ready. I think this is the most urgent aspect before turning multiomics into a reality in clinical practice.
And in this direction of the challenges,
I would just want to say something here just to follow up, and then I'll give you. Yeah, so I think right now multiomics is mostly being used as an exploratory analysis. For exploratory analysis, essentially, trying to identify mechanisms of disease. But to turn this into clinical practice, you need these machine learning approaches that, given a tumor profile, it's telling it has 67% probability of becoming disease A, 32% probability of turning into disease B, and so on. And this can only be achieved by, machine learning. Sorry, Aina, go ahead.
No, I was going to continue on these challenges because that's really relevant what you were saying, but also wanted to introduce what about other limitations in terms of maybe sample availability, because as you know, in clinical, how do you obtain the sample is something that you have to consider because resources are limited. Also, how invasive are techniques sometimes to get the samples. So also you have to consider this in your experimental design and how much multiomics we can do that, and if there is a way that we can prioritize some omics over others, or which perspective would you take on these regards? Also maybe regarding the budget limitations and experimental constraints. So how do you approach this thing?
Maybe Rob, can you start on that?
Sure. Yeah. So I think one sort of answer to that around sort of sample availability would be that in many cases we're using model systems, right? So part of my lab works on the development of brain tumors and how they can be actually controlled, and we're currently doing a study where we're looking at the Zika virus and whether that can be used to control the growth of brain tumors. Now, clearly that's going to be a very difficult experiment to do, in the clinic. So, we work with cell lines or we work with organoids and so forth. So model systems are really, really important here. But the limitations of model systems are just as they've always been. How much do they recapitulate actually what's happening, for example, in a patient? I think model systems are getting closer and closer. I mean, the last few years, development of organoids and other sort of multidimensional ways of growing tissues, for example, will be enormously important in the future. And being able to scale those up and then profile them using these different omics techniques, I think will be very, very powerful.
So I think there is an important point that Aina has raised here is which omic do you select to profile? Because as we know, we have budget constraints. We cannot profile every single molecular layer in a specific sample. So which ones do we go for? Now, I would argue most people have, large scale projects have mostly been focused on genetics on one aspect. UK Biobank is one of these great projects that allows you to try and relate genotype to phenotype. And somatic mutations are extremely useful. I mean, first because they are almost binary, and we like to work with binary things because they are easy to work with. But at the same time, this is like further away from the actual phenotype than most molecular layers, right? Usually you've got the transcriptome, you've got the proteome, you've got the epigenome, which tend to be, they probably explain more variation of your actual phenotype than genetics, although they are much harder to work with. So we need to bridge this gap to try and make sense of these intermediate data modalities, which are very hard to use in clinical practice. And yes, Aina?
No, it's going. Yeah, that you're totally right. It's quite complicated to get to this. So yeah, it's just like very appreciated, like how researchers are trying to integrate all this and try to figure out like which multiomics can be prioritized when it's something that from genotype to phenotype is not like the straight line of the biology dogma, right? From genome to the protein. So there's so many factors that can affect you, and not only about like the proteome, can only be like also the metabolomes that can make like some a little particle make like your protein active. So the sole presence of a protein doesn't give you a phenotype. So this gets like layers and layers of complexity, as you were saying before. And having said that, maybe I would like to ask you, because it's getting so interesting, that you could give more insights into your research in the single cell, like how are the biggest applications in this field research area, how are this is expected to evolve, right? Because you have some publications in terms of like chromatin accessibility, DNA methylation, RNA expression, but how is this like going to progress in future? What are your plans on this?
Yes, so in terms of the single cell area, this is a field that has done a massive leap on the last years, and I would say it probably started around 8 or 9 years ago, when for the first time we were able to profile RNA-seq in a large-scale fashion, so meaning more than one cell at a time. Now, it has grown massively, especially by efforts from some consortiums such as the Human Cell Atlas that I was mentioning before, where the aim is to create, sort of a reference map for each tissue on the human body. So, for a specific tissue that you profile, which are the molecular constituents of that tissue, which are the different cell types, and by profiling this in a series of healthy patients, you're going to have some sort of idea of what is the normal variability that you expect. And then if you profile samples from a disease context, by comparing to that reference, then you can tell, all right, there is a depletion of T cells, so there is a massive growing of B cells, and this allows you to get, what is the molecular constituent, what is the underlying condition of a specific disease. Now, in the single cell area, most of the effort has been focused on the RNA expression field. And it makes sense because, RNA expression is something which is fairly easy to work with in the sense that, we know how to interpret genes because we have good knowledge on how genes behave. We can easily perturb some genes and then assess the effect of that change in the transcriptome. But very recently, we're able to profile more than one molecular layer in one cell. So now I'm mostly working on a kit which was released by 10x Genomics, which is called the Multiome kit. And this allows you to profile RNA expression and chromatin accessibility from one cell. Now, how much are we going to be able to learn from that? This is still an open question, because if there are challenges on the computational aspect of standard multiomics, let's say, there is like 10 times more challenges when you work with single cell data, because it's extremely noisy, it's extremely sparse, and you cannot look at one gene at a time in a specific cell because it doesn't make sense. And here's where data science and machine learning again becomes really essential on how to interpret this data.
Sure, makes sense. Yeah, I think like you were mentioning now about like transcriptomics being very relevant. I think like at Novogene, right, Yuanyuan, like people get like more and more interested in like RNA solutions in transcriptomics, because at the beginning in this omics field, like first was genomics and everyone was in genomics, first whole genome sequencing, then whole exome sequencing, which complements and makes things much more feasible because like of the cost, and because like maybe 80% of the mutations were like supposed to be in the exome. But now more people are like considering the transcriptome, and I think it has like a lot of relevance in the multiomics field.
Yeah, I can maybe comment on this. As a service provider, we see a trend, as Anna mentioned, starting from genomic level, then into the transcriptomic level. And these recent years, we see more and more epigenetic level. For example, Ricard mentioned chromatin level or methylation level. And even more recently, we see even one more layer at the microbiome level. Actually, yesterday we had a co-session presented by Gianni, who illustrated how important is the microbiome contributing to the whole network, the whole system. We just find it very interesting that these multiomics keeps adding on top of each other and bringing more insight into all of the research areas. And this brings one of the questions from the audience. Actually, we saw that two questions. One question is, are there a favorite omics method on top of genomics? And the second question is, most of the multiomics are focused on cancer and autoimmune disease. Do you see the value of multiomics in other congenital diseases?
Yeah, I wanted to take the advantage that Rob hasn't replied like this last question to ask him if he sees like other diseases being addressed, because like as Yuanyuan mentioned, like all the focus seems to be in cancer, and you yourself like are quite familiar with this. So do you have any expectations on how this will develop in terms of the multiomics?
Oh, absolutely. Yeah, I think one obvious, very topical area would be infectious disease. So, some of our work focuses on how viruses interact with their host cells. So, that obviously leads to a kind of proteomic type approach. So we look at the protein-protein interactions between viral proteins and the host cells. But if you want to get to the dynamics of that process, then other techniques such as RNA-seq can be very, very valuable. So you want quantitative information because obviously the interaction of the virus with its host is a very dynamic process. So I think infectious disease is going to be really, really, really important. There's been quite a lot of work in the COVID field in terms of looking at protein interactions. Other people are looking at the host response using, for example, transcriptomics and quantitative proteomics. So this is only going to grow, I think. So I see infectious disease as one of the things where multiomics will have a real contribution on basic understanding of how these pathogens are actually working.
What about you, Ricard? Any other thoughts?
So I think that any disease that doesn't have a clear genomic target, that it's not a Mendelian disease that we have identified a specific gene that drives that mutation. So any disease that has heterogeneity between your patients that cannot be pinpointed by a specific mutation is a potential target for multiomics. Because then that means that if the genome doesn't explain it, there has to be some changes in some molecular layers in your cell, which are the drivers of that disease. Now we typically
Definitely.
Go on, Aina.
No, sorry, I was just going to say definitely like the more complex one disease is, the more can benefit from these different layers that multiomics can offer. So
yeah, exactly. So I think multiomics is a strategy to address complexity. So the more complex your system is, the more you will benefit from getting a multiomics.
And how do you see the short-term and long-term future of these multiomics, and how consortiums and data repositories and the whole field will evolve in these regards? Do you have any expectations, or how in your own experience think that these things will go? So maybe Rob, you can go.
Yeah, sure. Yeah. So I think it's only going to grow, right? I think that all the things we've been talking about here, point to sort of the growth and one thing I think we need to think about is sort of accessibilities to technologies. So if people come with biological problems, how accessible are these different technologies? And as we pointed out here, and as Yuanyuan pointed out, there are really nice service providers that can allow us to get good data on these systems. But accessibility, perhaps in more emerging omics techniques, might be more limited. So I think that's something that needs to be addressed to fully benefit from multiomics.
It's maybe more like now the strategy we follow rather than the cost of it, since like we're having like so many emerging technologies and so many resources that could be integrating data or just providing a bigger data set of data. Should we more put the effort into trying to combine the data rather than trying to just produce? Like how do you think this will evolve in this case, like Ricard, from your perspective?
I think in that case, and it's in line with what I said before, that consortiums are important in that aspect, and because doing a proper multiomics, it takes money, it takes effort, it takes time, and in some way the UK is really a powerhouse on this. Just to describe the biobank, the UK biobank example, I think they're mostly focused on genomics and also RNA expression for now, but they are doing this on on the scale of thousands of patients. And this is unmatched by any other country. And this is exactly the type of resources that are going to allow us to take multiomics and then applying it in real like clinical cases. I suspect that what the UK Biobank will do in the end, and other consortiums should follow up, is we start from the genome, we start from the transcriptome, but then we need to start adding. They will likely freeze some samples, but then eventually they will decide, all right, for some reason, for that specific disease, methylation might probably be very important. So let's add some methylation samples, and then let's see how much do we actually gain by including this third layer. If we don't explain anything by methylation, that means we'll likely need to turn into another omic, like other type of epigenetics or the microbiome or the metabolomics. And this hierarchical approach is what I think will allow us to take multiomics into clinical practice.
Yeah, I mean, I think definitely agree with what Ricard's saying there. I think the big consortia, I mean, if you look sort of, what was it, sort of more than 10 years ago, actually, when the NIH decided to invest in the Cancer Genome Atlas, that's absolutely revolutionized multiomics. So essentially taking large cohorts of diverse cancer types and tumors and profiling them in different ways was massive. And it was massive in terms of both discovery, but also driving the development of tools and accessibility. So those things are going to remain really, really important. There's going to be kind of cottage industry type projects where individual labs using multiomics, but I think big consortia that are processing samples and generating data in a standardized way that's reproducible and validated and so forth is going to be hugely important to drive the field forward.
Yeah, definitely. That will be probably the case. So having said that, Yuanyuan, do you have more questions from the audience? Can you see them?
I think
maybe we can throw some of them.
The 2 questions from the audience.
I can see one now here. So yeah, it mentions about like how to use the data to optimize pipelines. So maybe it's a bit like what we already mentioned, but maybe you can give a deeper insight into that. So how would you update or optimize the pipelines you're currently using for processing your data, or which tools are you right now using for getting a more multiomic approach to your questions?
I guess I can jump in there and sort of say that, from the proteomics perspective, it's been really important to try and sort of validate what we're getting, and using things like sort of spike-ins or synthetic samples is the way to go there. To actually really sort of validate calibrate the instrumentation and make sure that your pipeline is giving you valid data.
Mhm. Good. Also, we have another question on, so speaking from machine learning aspect, we think that the challenge is to have access to data that we could be potentially integrated and then to advance the integration across multiple omic data sets. So what's your opinion on that? Maybe Ricard,
yeah, absolutely. I mean, I think that data is the main limitation when it comes to any sort of machine learning approach. And as we were saying, like there are different labs doing like multiomics, but if one lab has processed 10 multiomic samples, another lab has processed 10 multiomic samples using completely different protocols, with different different labs, different strategies, then integrating this data into a unified framework, it becomes much more challenging than if it's one single consortium that builds up 1,000 samples from cancer X, right? Then make sure to unify and like reduce the amount of technical variability that you see in your sample, and this is what allows you to build proper statistical methods around that. But I was saying, for multiomics, I think one of the key aspects is the analysis aspect. Not only in terms of like batch correction methods, meaning that you're going to get a lot of technical differences between all your samples, and once you remove that is when you are able to start doing some data integration across all your different omics, and this is a complete other field that we can discuss.
And talking about machine learning, do you think that the data we have right now or the future designs for experimental multiomics, so is it important to see if there is a bias in our data? Because maybe you when we design or when we take samples, it's more like from a Western perspective, so that our data sets are biased and the conclusions we can take right now in these fragmented data sets are not totally accurate. And so we should make an effort on that, not only at the single omics, but also at the multi-omics perspective.
I think there's undoubtedly bias, in the samples we're looking at. I mean, just to actually, Anna, just to follow up on what Ricard was saying, I agree entirely. I think the consortium approach where you're validating samples, perhaps you're exchanging the same samples between labs, ensuring that, for example, instrumentation is giving the same output is going to be really, really important. I think it's really, a question here is it's really important for us to understand what data can be integrated together. I think that's a question that we haven't fully sort of answered. When is it appropriate to integrate data, and what conclusions can be drawn? The boundaries, as it were, around that question are not well defined, I don't think.
That's right. Also, another question that comes up is, so as we were discussing, multiomics is getting more and more attention recently. So do you anticipate a major switch towards multiomics approaches? Well, clearly we mentioned, yeah. So, and they would like you to highlight again, like what would stop the bottlenecks. So which are the bottlenecks that we have to definitely surpass to have like a more proliferant multiomics?
Yeah, I mean, I think we've discussed this. The main limit, like methods are out there, but we need to do benchmark on those methods, for example. And to develop new methods, if you don't have the data, there is nothing to be trained on, right? So, and there is data already, and models are being used, are being like developed. But ideally, we need more multiomics data from large scale like consortiums, and I think like both Rob and I agree on that.
That's good. So, well, I think like now maybe we are reaching to an end into this discussion for time restrictions. So I would like to thank you all for participating to this mastermind session today, and for all of the attendees and their questions. So thank you, everyone. It's been a great pleasure to be discussing about this topic.
Thank you very much.
Thanks a lot. It's a
Thanks,
pleasure,
Aina.
and I think it's very, very, very nice topic.